Who can read this? Well, would that be better? And because you know the English language, you can make sense of this text, and you could recognize that there was a problem with the previous one. How about this series of A, C, G, and T that represents a DNA sequence? Well, I don't know about you, but I don't speak DNA, so it's really very difficult to figure out if this sequence is correct or not, what it does. And so because of that limitation, I structured the research program of my group really after a language acquisition process. We spent a lot of time listening to DNA and genomic data. We try to make sense of it. When we think that we have all figured it out, we make a synthetic DNA molecule, put it in a cell, look what it does. And today we are going to be talking mainly about the process of trying to figure out what it does and designing it. And before we dive into that, we have to keep in mind that the theoretical framework of molecular biology is really built on a linguistic metaphor. Notions of DNA replication, transcription, translation are all based, genetic code are all based on this analogy. And this analogy is 50 years old. And what we are trying to do is actually try to formalize it, to push it further using the theory of formal languages. So formal language can be seen as a series of rewriting rules. So for instance, if I want to write a sentence, I know that the sentence needs to be composed of a subject and a predicate. A predicate would be a verb and an object, and so on and so forth. So now if I want to write a sentence, I'm going to apply all these rewriting rules successively until I reach a point where no more rewriting is possible. At this point, I can collect all the leaves of this derivation tree, and that gives me the structure, a template for a sentence. I can pick words in each category, and that gives me an actual sentence. And if I iterate that process, I can write Hamlet, more or less. So we can try to do the same with the process of designing a synthetic DNA molecule. I'm going to apply a number of rewriting rules that will, shape the structure of the DNA molecule until the stage where no more rewriting is possible, and then I'm going to choose some genetic parts of the different categories to get the DNA sequence of the new synthetic DNA molecule. And in this case, that's a genetic switch. So far, we have tried to develop a framework to make sure that the DNA molecule we make are structurally valid, but that's not enough. We have to go beyond that syntactic level to try to figure out what the DNA molecule is going to do. And in order to do that, we have to keep in mind that words don't really have any meaning by themselves. So for instance, bear claw can refer to a body part or a pastry, depending on which context it's used. And similarly, the DNA sequence coding for a particular protein called gene really is going to have a very different effect on the cell, depending on the genomic context in which it's inserted. So we capture all these context dependencies in a formalism called attribute grammars. And this formalism makes it possible to translate the DNA sequence into a dynamical model of all the molecular interactions that are encoded in that DNA sequence. And this compilation step is really a prerequisite to a systematic exploration of the design space generated by a library of genetic parts. And we can explore this design space automatically and try to find an optimal design that maximizes some figure of merit. Looking forward, we can anticipate an evolution of DNA programming languages that is going to be very similar to the evolution of computer programming languages. Most molecular biologists today work at the DNA sequence level, which is very close to the machine code. The languages that I showed today could be compared to assembly languages, but looking forward, we would really like to have a third generation of portable languages that would make it possible to compile a single genetic design for different target organisms, a DNA sequence for plants, a DNA sequence for mice, for instance, just like you can compile a C program for different microprocessors. All of this theoretical development would be really pointless unless we can package it into an application that's usable by our target user group, which are molecular biologists. They are not engineers, they are not computer scientists, and they don't know any of that. So that's why we have developed a web-based open source application called GenoCat that's available at genocat.org, and I encourage you to have a look. Thank you.